Hello all, welcome back to the physical evidence classes, and I am Dr. Bhoopesh, your forensic tutor. So in the previous session we have discussed about the significance of physical evidences, types of physical evidences, and location and the source of physical evidence. This time we are going to discuss about the collection of physical evidences. What are the different measures we need to take, what are the different precautions we need to take during the collection of the physical evidences on the crime scene. Now as you can see here, the first thing which says any temptation of picking up an article immediately on arrival at the scene should be avoided. As an investigator or as a forensic expert, you might be excited to see the physical evidences or to jump off over the physical evidences to pick them up immediately. So you need to collect them, you want to collect them and you need to send them immediately to the lab. But no, it is not the method. There is a proper searching method, there is a proper collection method. So any temptation, any such excitement should be avoided. Okay, otherwise we will misplace the evidence or we will destroy the evidence in this case. Now number second is all the evidence should be collected after the initial search and recording of the crime scene. Recording of the crime scene we will discuss later on in detail. Recording by photography, recording by sketches, recorded by notes. So once these all three things have been done and after the initial search have been done, then only the evidence should be collected. Okay, let's say if you are collecting the evidence right now without the proper recording, and if you have misplaced the evidence, so the integrity of the evidence is lost. So you need to take care of that. You should not collect it immediately, but after the initial search and proper recording of the crime scene. Third thing is all the evidence should be collected with all precautions, means wearing the gloves, your kits. Okay, even there can be some evidences which can create infections, so you need to prepare yourself also. You need to be precautious as well. While do not touch anything directly, otherwise there can be chance of transferring your DNA, your sweat, your fingerprints onto the other evidences. So please avoid that. And also there are chances of destruction, okay, or contamination, cross contamination. So you need to take care of the proper collecting kits and proper collecting containers as visible in point number 4. All the evidence should be collected in separate container. What does that mean? Let's say there are 3 pools of blood. 3 blood pools are there and you need to collect them. Do you want them to collect in the same container? No, because they may be from 3 different sources. You do not know, or one may be contaminated. So we need to collect them in 3 separate containers, airtight containers, and we will pack them. We will label them as blood sample 1, blood sample 2, blood sample 3, or whatever the requirement of that forensic lab is. We will label it according to that. Now suppose if there are 3 cartridge cases, so you will say that there is no chances of cross contamination of cartridge cases in together. No, there is, because if the cartridge cases will collide with each other, they will create extra markings. Now these extra markings will create a hindrance in the identification of the individual markings over the cartridge case. So everything should be collected in a separate and appropriate container. Next is in violent crime. We are talking about the crime where the sexual assault has happened or the murder have happened. So in such crimes, the evidence should be collected before the removal of the body, because after the removal of the body you may get more evidences beneath the body. Then they should be collected with proper method in proper collecting devices. I will tell you about the collecting devices later on in the further slides. So because we need device and containers properly according to the evidence. Let's say if there is a hair, if I ask you how you will collect hair, and if you will say we will collect hair in a big jar, okay, so what may happen? You may lost it the while during the parcel, or when it will reach to the forensic scientist in the lab. What may happen? He may overlook that, or he may think that jar is empty. So hair should be collected in a white envelope. If it is black hair, it should be collected in white envelope. If it is white hair, it should be collected in black color envelope or contrast color. envelope. So proper container and proper collecting device is very much necessary while the collection of the evidences. Number 7 is they should be handled properly. Now here handling means you should not drop anything once you picked it up. Okay, so you have to be very careful. So you need to avoid any loss or any damage of such kind. Then you need to maintain the chain of custody. I think you all know what is chain of custody. Whosoever is handling the evidence one after the other, they need to sign, they need to keep a record of that. So this is chain of custody. For example, if I am the investigating officer, I am collecting the evidence, or the forensic expert is collecting the evidence, he will give it to me, I will sign, he will sign with the date and time, and I will give it to the another expert or another person to take it to the forensic lab. He will sign and I will also sign along with the date and time. So the integrity of the evidence can be maintained, and if there is any mishap happens, so we can say who is responsible for that mishap. Okay, so let's move ahead. Packaging and labeling. Now packaging is quite similar in all the countries, but labeling can be different in different parts of the world. What has to be mentioned on the labeling, but most of the things are quite similar. Okay, so the packing should be proper and secure, means there should be no chance of destruction of that packing, or if somebody may open it and reseal it, it should not happen. So sealing should be done, securing should be done, as like if anybody opens it, he should not be able to seal it or close it in the same way as it was closed. So you can check whether or otherwise it has been opened or not. Okay, second thing is each article should be separately packed and labeled so that individuality and identity are maintained, as I told you before in the previous slide as well. Next is, as far as possible, all packets belonging to one case should be enclosed in a box. Now you will say, sir, you were saying that every item should be collected in different container. Now why in one box? No, we are talking about big carton, big box. Okay, so all the small boxes can then be placed in a one big box, and then that big box can have all the details of the case number, case type, and the investigator details, the forensic expert details, all the evidences, exhibit peg, all these things can be there on that box. But there are certain evidences, for example, dead body is there. Dead body cannot be packed in that box, so it will definitely go separately. And also a firearm is there. Firearm will not be packed in that, so it will go separately. If a big firearm, I am talking about rifle or so, you have to pack it separately. But as far as possible, try to do it in one packing, and you have to avoid the cross contamination also. If there is something which can create a cross contamination like blood or semen or any other biological fluid, so you should be careful to pack it completely, so you should not cross contaminate the other evidences as well. All the exhibits inside the packing do not come in contact with each other for the cross contamination that we know. The exhibit do not spill due to the, so if you are having the lids, if they are not properly tightened, or the stoppers, if they are not properly tightened, there may be chances of spill due to that loosened lid. So what you need to do, you need to tighten it. Wherever it is required airtight container, you need to keep airtight container. Wherever it is required ventilated container, then you have to use the well ventilated container, paper envelopes, all like this. Okay, we will talk about that also. The container should be strong enough to avoid any damage in transit. So if you are using glass vials or glass jars, you should be very careful. So try to use the containers of steel and plastic as far as possible. Okay, so next let us move to another slide. The labeling should be numbered. The label should be numbered and should be at the signature of the forwarding officer, that you all know. We have discussed the chain of custody. Okay, the top side of the box, if you are packing that in a one big box, so top side should be marked with an arrow, that which is the top side. Otherwise anybody can hold it from the different side, opposite sides, top down. Okay, so everything will be spoiled. So the top side should be mentioned clearly with an arrow or with a mark. Okay, so anybody will handle it properly. The exhibit should be sent through a special messenger as far as possible. No, it is like that. You should not send it by courier or by parcel. You should have the personal officials to take those evidences to the crime lab, and the evidences should be properly sealed. Okay. Now next is, let's see. Sorry. So for minute traces like hairs, fiber, dust, soil, glass plane, vacuum sweeping should be done. Now this vacuum is not the vacuum cleaner what you have in your house. These are the micro vacuums, and you need to change the vacuum pouches every time. If you are collecting one evidence, change the pouch again, then collect the other evidence, change the pouch again, and so on. Okay. It may be necessary to take the custody of all clothing worn by the participant in a crime. So, because clothing may have certain evidences in them, and you may have to sweep or vacuum the clothing as well for minute traces like hairs and soils, hair and soil. Okay. Now, fingernail scrapings. If there is a sexual assault case, so you may have the tissues in fingernails. So fingernails of the victim. Okay, so they should be scraped out with a blunt instrument, not with a sharp, because if you will insert a sharp instrument, there may be chances of bleeding, and that blood will mix with the tissue of the culprit or the perpetrator, and there will be cross contamination. So like a toothpick can be used, a spatula can be used, like that anything can be used, and they can be subjected to microscopic examination further. Now these are the types of the forensic vacuums which you can see on net as well further. So these are the type of the micro vacuums which we use, and the pouches are being changed separately. Again the micro vacuum. These are different forensic vacuums. Now next is, we are talking if there is a deceased victim. The body will be autopsied by a medical examiner or coroner to collect the evidences. Let's say there are certain evidences which you can see over the dead body. There is an injury mark, so you can photograph it at the crime scene. You cannot collect it. It will be again photographed by the medical examiner or the coroner in the autopsy room in the mortuary, and then the swabbing will be done by him. You should not try to swab it out there itself. Let's say there is a froth coming out from the mouth of a person. What you will do? You can swab out the extra froth which is coming out, and you can collect in a separate container, and then again mention in your report while collecting the evidences that there was a froth beside the mouth of the victim. It may be a poison, it may be a symptom of a poison, a toxin material. So again it will be collected properly, swabbed out properly by the medical examiner. The second thing is, they will be working to determine what the medical examiners do. They will determine the cause of death, the manner of death, and the nature of death while collecting the evidences. Okay. Now all the dried stains on immovable items. There are two types of items. Movable, let's say a small chair is there. You can easily seal the chair, or you can easily send the chair to the forensic lab. But if there is a blood stain, a bloody palm stain on a wall, what you are going to do? You will cut the wall? No. You will photograph it first. See, all the collections have to be done after proper recording. I told you earlier also, after proper recording. So consider you have already photographed it completely. Now you have a bloody palm print on a wall. You can swab it out for the blood analysis. Okay, because you have already taken the photograph for the fingerprint and palm print impression. Now for this, cotton swab moistened with distilled water or saline water can be used. These are separate containers, the vials which you can use in some cases like that. Okay, so these are the containers which can be used for large blood pools, vomit materials, fecal materials, tissues, lungs, liver, or anything like that. Now these can be used for swabbing or needles, and for electronic wires and all. For tools, you can make a tool like this. For clothing, paper envelope should be used, and these are the small vials for small swabbings, or maybe for cartridge cases as well. Okay, for firearms, you need to use the appropriate size cardboard box. If there is a long firearm, you need to use long cardboard box. If it's a short firearm, you need to use short cardboard box. And you need to tie like this. Why? Because if it will move, if it is loaded, if it is malfunctioning, so it may misfire again, there may be accidental firing. Secondly, if it is having a fingerprint on it, it may get damaged. So we need to tie this the firearm like this. And the mobile phones, because of the fingerprint, if we will keep them in a hard container, the fingerprints can be wiped off. So the paper envelope can be used for the same thing. And again, this is the bigger tool, the same type which is mentioned here. Even the knives can be put like this with a cork inside. The sharp edge of the knife should be inserted in a cork, so it will not damage the other evidences. If there is any trace material like blood, hair, fiber, or soil particles, they should not be removed from any garment. Instead, the entire garment or entire object should be sent to the crime scene lab, because they may have certain other evidences as well, which may be very beneficial in the later stages of the investigation. The second thing is liquid biological fluids. Okay, again we are talking about the blood, semen, saliva, urine, tears, all those things. They should be collected with the help of the droppers and pipettes. If there is a pool and the blood is still wet, they should be collected with the droppers till the dropper is allowing you to collect. And once there is no more blood for the dropper, then you can apply swabbing. Okay, and then you can put them in appropriate vials or the swabbing vials. Then these are the types of the details which should be there on the chain of custody chart, or the proper labeling, you can say. If the evidence is found adhering to a large structure such as door wall, remove the specimen with the help of a forceps or appropriate tool. Let's say in the case of blood stain, one may either scrap it out. If there is a small blood stain on the wall, you can scrap. Still wet, they should be air dried first at the room temperature, and they should be collected in a ventilated paper bags. Okay, so please remember this. Wet stains should be air dried at room temperature. They should not be dried with the help of a hair dryer or anything. Otherwise the DNA can get degenerated or fragmented. So we need to avoid warm drying. We need to dry it at room temperature. We need to wait for that. Now all the volatiles like petrol, alcohol and others, they should be collected in airtight containers. Okay, these are again some of the examples I would like to mention here. This we have already discussed, that the firearm should be collected in appropriate size cardboard box. Okay, the tools with the help of a cork, so it will not damage the other evidence, and it will not collide with the walls of the container as well. The hair should be collected in a paper envelope like this, but as far as possible try to use the contrast color paper envelope if available. If not, then it is same, then you are going to label it that it contains this color of hair. Examination. In case you are collecting the cartridge with the help of the forceps, if you will touch this side of the forceps at the top of the cartridge, it will create extra marking which will play a hindrance in the proper identification of the cartridge case. So what you need to do, you need to insert it inside and open the forceps and then collect it in a proper container. You can use a matchbox with filled with cotton for the cartridge case and bullets as well. Okay, and the last is how to collect the standard reference samples. Now, you know, forensic science is all about the science of comparison. So law of comparison is regulating there. So when you have the crime samples collected from the crime scene, you have the specimens that have to be collected properly from the suspects, from the suspect surroundings, etc. So the same rule should be followed in collection of the standard reference samples, and we will learn this further in detail in the slides ahead. For now, thank you very much. So this was the physical evidence completed. Next time we are going to start with a new chapter. Thank you.